Now let's move into section 2, where we are going to talk about prokaryotic cells. Our learning objectives here are to name examples of prokaryotic and eukaryotic organisms, compare and contrast prokaryotic and eukaryotic cells, describe the relative sizes of different cells, and then explain why cells must be small. So first, let's talk about the components that make up prokaryotic cells. If you remember, the word prokaryotic really refers to us not having a nucleus within these organisms or within these cells. So it is the simple and mostly single-celled, also termed as unicell, organisms that are going to lack that nucleus or membrane-bound organelles. And we'll get into a bit more detail on what organelles are in other chapters, but also we'll mention them here and there in this lecture too. So in our prokaryotic cells, we're going to have a plasma membrane just like we will with our regular animal cells. So it's really that boundary around the cell that is going to have that semi-permeable membrane and be the gatekeeper for what moves into and out of our cell. We also can have a peptidoglycan cell wall in these bacterial cells, where we may have more compounds that will make up a thicker peptidoglycan wall or a thinner peptidoglycan wall, and we'll talk about these differences more within a microbiology course. And if we have a bacterial cell that has a special virulent factor, which I know that term virulent factor may be new to you, but I like to think of it as different tools that these organisms have in order to create damage within our body or evade or kind of hide out from our immune system. So the capsule, I would say, is something that helps us hide from the immune system. Basically, it is a slippery, slimy wall that when we have certain immune cells that try to grasp this bacterial cell in order to eat it up and basically destroy it, instead, it cannot grasp onto it. Think about if you are trying to hold on to a slippery bar of soap. It just keeps slipping out of your hand. And so in this way, it's a tool to stay alive, right? And then we have our cytoplasm with our cytosol, which is going to be that fluid that we find from the plasma membrane to well, really all throughout the cell, and we can kind of hold its genetic information within it and other organelles that it has. And our genetic information I mentioned will be found in a region called the nucleoid region, and our ribosomes have to be there in order to synthesize or make proteins. And lastly, some of these cells are going to have flagella, pili, or fimbriae. So flagella is going to be this long structure back here, and it really helps with motility or movement of the cell. Pili, we don't see, oh, we do see a little bit of it pictured over here. These are extensions that move out of the cell and can connect to another cell in order to transport genetic information. And that's through a process called conjugation. Again, we'll get more into that into a microbiology course. And then fimbriae are also going to be little extensions off of the cell, and that really helps with adherence. It can basically attach to different structures, whether it is our mucous membrane or other cells. Now we have our cell size. So our prokaryotic cells are typically going to be much smaller than our eukaryotic cells. So you can see this figure down here that we are able to compare some of our prokaryotic cells to the size of our eukaryotic cells. And typically the smaller an organism is, the more easily we'll be able to have structures enter and exit the cell, so we can more easily get nutrients and get rid of waste products. Whereas when we have something that typically is more multicellular, like our eukaryotic cells, we need more mechanisms of transport. So more on that later on. Now we know that our cells are really small and we have to use a microscope to view them. And the reason they're so small is because they actually are going to have a surface area to volume ratio that is going to make the cell more efficient. So our smaller cells are going to have a larger amount of surface area compared to its volume, which will allow it to have more exchange of nutrients and wastes. And there is a limit to how large a cell can be while remaining efficient and metabolically active. So if we have a cell that grows too big, it's going to become less efficient. And it can, though, start to increase its surface area by folding and increasing its surface area. And so we can see a good illustration of this up here where we have a cube. This can represent our cell. And notice how much surface it has over here. We've got all of the numbers listed below. And then when we move to our 8 2-centimeter cubes, we can see that we've now increased surface area because now we've got surfaces all in between inside of this cube. And when we increase that to 64, we are going to definitely highly increase that surface area even further so that these cells can be more efficient in their exchange.